It is a real pleasure to present to you today work that we at Kids First, alongside with partners at GTEx, LINCS, the coordinating centers, and others across the CFDE, have been doing on behalf of empowering platform-based approaches for accelerated discovery and clinical translation. A central challenge for every DCC and for platform-based ecosystems as a whole is the creation and deployment of tools and resources that ultimately accelerate the capacity of end users to find and identify large-scale datasets that can be rapidly converted to structured information that can be harmonized and then leveraged for new knowledge creation and impact on behalf of patients and their families. To guide the development of platform-based approaches across DCC datasets, we sought to utilize real-world use cases from the Kids First cohorts of structural birth defects and pediatric cancer. For today's presentation, we'll highlight one such use case from Dr. Maris and Dr. Diskin focusing on neuroblastoma, a pediatric tumor of the adrenal gland. A shared unmet need across all of pediatric cancer and rare disease research is identifying new targets that are unique to the tumor or affected tissue, such that therapies will not affect normal or other developing tissue. Such efforts in this Cancer Cell manuscript by Dr. Diskin, Dr. Maris, and their colleagues sought to do this in 2017 by downloading existing GTEx data, combining it with their own neuroblastoma data, and performing such analysis in order to identify novel targets, including GPC2. Such effort typically represent a multi-year process for investigators to identify the data that they are interested in across resource, convert that data into harmonized information, to begin a process of knowledge creation and ultimately translational impact. Through CFDE-based collaborations, we sought to address these challenges in ways that would be extensible across DCC's other datasets on behalf of future research. Cohort creation is empowered for investigators through the Common Fund Data Ecosystem Search Portal, where investigators can make searches across CFDE DCCs for datasets and subjects of interest. However, a key challenge in the data phase of discovery is the coordination and assembly of these datasets across DCCs in a meaningful way that can be rapid and accelerate the conversion of data to information by investigators. To address this, we partnered with the CFDE portal developers to implement additional resources that leveraged NIH Cloud Platform interoperability efforts across RAS and DERS in order to support the manifest creation for workspace deployment. Here you can see a user leveraging the CFDE portal in order to select GTEx and Kids First datasets focusing on RNA sequencing assays in order to create a manifest that they can export and utilize within a cloud workspace environment, leveraging the NCPI file manifest standards. With manifest in hand, a user is now able to begin the information phase of the discovery process by leveraging the manifest in a compliant workspace like Cavatica. Importantly, Cavatica and other such workspaces can interpret the requisite dbGaP permissions for controlled access or open access and allows a user to integrate their data with their dbGaP permissioned datasets across GTEx and Kids First. Here, a user is taking the CFDE manifest, uploading it into the Cavatica workspace, and in doing so, the Cavatica workspace will simply point to the local storage environment of those data, obviating the need to create a secondary copy of those data on behalf of the user. With data now in hand in a collaborative workspace, a user can choose to run their own pipelines on behalf of harmonizing such data, or can leverage any one of the CFDE pipelines already incorporated into the Kids First environment on behalf of harmonizing data across Kids First and or GTEx data. The user is now ready to enter the knowledge phase of discovery. After running such pipelines, a user will end up with a large number of files per sample as outputted from each of the workflows. Oftentimes, such files must be restructured in the context of matrices or tables in order to engage further downstream analysis and interpretation of such data, bringing together the multiple files across the datasets into a common view on behalf of the user. However, as part of the CFDE efforts and on behalf of translating knowledge to impact, we sought to empower users' capacity to recapitulate analysis in a more easy and facile manner using Apertors in partnership with Lynx and Avi Mayan's group. Here you see an Apertor specifically designed to do the exact same analysis as highlighted in that Cancer Cell paper reviewing neuroblastoma and GTEx. Importantly, the Apertor can actually interoperate with and connect to the Kavatica workspace where data were processed, harmonized, and structured. A user can very rapidly begin to review a set of genes that have been identified as differentially expressed across their own data, GTEx data, and the Kids First neuroblastoma dataset. We check to see if this new analysis using the Apertors recapitulate the original findings of the Cancer Cell paper from 2017 with this new larger cohort of neuroblastoma. Indeed, many of the same genes that were initially identified were also identified as part of this analysis via the Apertor. Additional genes were also identified, including FOX2B and FOX2A, genes that were not initially in the list from the cancer research paper. The value of this end-to-end approach and the additional findings by the Apertor were recently validated by additional publications by Dr. Maris and his colleagues, where they did indeed add to their previous list of targets by performing additional analysis, including proteomics, and then likewise, again, leveraged GTEx for comparative analysis, and as can be seen here, identified FOX2B as an additional target. We hope this presentation provided you a view of the potential impact of the Common Fund data ecosystem and the resources being enabled across teams and DCCs.